So let's go back here. Okay, so I'm recording this right now just for future references. Again, if there's like a field trip, a sports game like team thing, or like a music performance, that kind of stuff, where you're going to miss classes ahead of time, definitely email me to request the lesson to be recorded. Typically, because of we have two lessons back to back, period 1 by 11 bio, period 2 11 bio. When I'm recording a lesson, I'll be recording the second period class. So if your first period, I'm looking at camera, if your first period students and you kind of like, why is Mr. Cheng not recording my class? That's a reason why. There shouldn't be too much material difference between both classes. I try to make sure of that. So that's that. So in terms of today, we've gone through the homework. The link to the answer is here. Now we're going to talk about protists. Okay, today we're going to talk about protists, which now this lesson about protists, in some ways, it's the first of the eukaryotic kingdom. Okay, we've been talking a lot about prokaryotes like eubacteria and archaea. We spent like a lesson and a half or 2 lessons on viruses and immune response and immunization. So today we're going to talk about protists. It's the first of the eukaryotic kingdom. Okay, and in some ways it is a very, very diverse kingdom. Okay, it is a very diverse kingdom. It is very small. It's very diverse. Now, one teacher that I worked with before, he would actually call this kingdom, he was British, so he'd say, it's like the dustbin of eukaryotes. Basically, if you cannot classify it as fungi, plants, or animal, and it's not a prokaryote, they'll just shove it in there. As a result, it is very, very diverse and it's very, very different. Okay, some are single cells, some are multiple cells, some have cell walls, some don't. Some make their own food, autotroph. Some need to eat some other things. So it's very, very different. Now, if we look at the if we look at the evolutionary history, okay. So we first started having our first eukaryotic cell. And then from what is believed, as evolution goes, as you can see here, it branches through, the phylogeny tree branches through a number of times before we get to even plants, fungi, and animals. So according to evolutionary history, these 3 kingdoms, animal, plants, and fungi, are relatively late compared to the eukaryotes, compared to the protists, okay? Because as you can see here, protists are basically, if it doesn't fit into these 3, it'll fit it in there, okay? Yes?
Because all these are like, besides like plantae, fungi, and amelia, why aren't they considered kingdoms?
These 3 are kingdoms. I'm not sure. I think they're certainly trying to limit the number of kingdoms there are. And I think they are limiting the number of kingdoms there are. And if we look back in terms of history, the first kingdoms there were like originally plants and animals, because they weren't microscope. And then later on, people realized, oh wait, fungi are not the same as plants. They separate. And then you have microscope. And it was like, oh, wait a minute. You have all these microorganisms. So in some ways, the oldest kingdoms have always been animals, and when I'm saying oldest, I mean human classification-wise, these are the oldest kingdoms. The later on ones, they just kind of put them all together. I don't know why, but I guess there's also a certain aspect of you don't want to have like 15 different kingdoms. Okay. That's a good question. Okay. Now, in terms of, so in this case, Protist is believed to be the oldest eukaryotic kingdom. And so in some ways, how do we get protists? Oh, no, how do we get eukaryotic cells? Yes?
Quick question. For the phylogenic gene, which one's the protists?
The what?
Which branch is the protists?
All the yellows.
Oh.
Okay, so where did eukaryotic cell come from? Okay, so in this one, you have two different theories. So you start off with the regular prokaryotic cells. Your DNA is spread around. There's no membrane brown nucleus. And then somehow, some of these plasma membrane or the cell membrane start caving in and form the early nuclear membrane, as well as some of these endoplasmic reticulum. Okay, that's one part of it in terms of the formation of the nucleus. But in terms of the other parts, predominantly your mitochondria or mitochondrion and chloroplast. This part here, there's a term in the theory called endosymbiosis or endosymbiotic theory. So, oops. Okay, so. Oh, I don't want to print anything. All right. So please write down these things here. I'm going to give you some time to copy this down. Okay. By the way, all the videos, if I record the lesson, all the videos, the link will be found on the unit planner and the videos will actually be on YouTube.
Mr. Chang, what does it say over there in the brackets underneath mitochondria?
Here?
Yeah.
Oh, this here is lowercase pl. This is SING because mitochondria and mitochondrion, that's a Latin-based name. So mitochondrion will be singular. Mitochondria would be plural. Okay. You're welcome. So in this case, like when you're reading this, it's like, why is Mr. Cheng writing mitochondria when here they talk about mitochondrion? It's the same thing, just plural and singular. Yes.
So technically, like the research and science that falls under like the category of endosymbiosis, would it just be generally biology or like
It would be a part of, like, it will be within the big umbrella of biology.
would there be like a sub classification for it specifically on like
I'm guessing that would be part of the evolutionary biology. That's my guess. That was not my major. Okay. So in this case, this theory, endosymbiotic theory or endosymbiosis, now just use a little bit of the words, kind of break down this word. Symbiosis or symbiotic. What does that mean? Symbiotic, symbiosis or symbiotic? Yes. Pardon? At harmony with each other could be.
They both benefit from each other.
That's a type of that's a type of symbiotic relationship. Do you guys just curious? In grade 9, did you guys learn about like how like different in your ecosystem unit? Did you talk about like how different species interact with each other? So some relationship will be like mutually beneficial. That's called mutualism. Some are like one benefit, the other one is like neutral. So it's called commercialism. And then one, it's harmful to another one, where you will call it either parasitic, or if it's outright kill to the other one, it will be not murderous, not carnivore, predatory. Okay. So do you guys remember that term? So if you remember, symbiotic relationship will be like the relationship between two different species. Endosymbiotic theory or endosymbiosis. Now, how does it? We're really talking about how these cells that used to be prokaryotes now gain these organelles. So I'm gonna just go back to the colored, nicer picture. I'm just gonna hide this. So essentially, the theory goes one day one larger prokaryotic cell, former prokaryotic cell, have engulfed another smaller prokaryotic cell. But rather than breaking it down for food, as it usually might do, for some reason, we don't know why, the larger cell decides, like, you know what, I'll let this guy live. But then they will have a mutual beneficial arrangement. So the bigger guy will provide food and shelter to the smaller cell, and in return, the smaller cell will provide energy by breaking down the food that the larger cell provides into a usable form of energy, like ATP. Okay, so that's kind of like that's how the theory goes in terms of how eukaryotic cells start having like mitochondria. So in some ways, for all the eukaryotic cells that need to eat other things to live, like heterotrophic eukaryotic cells, that's kind of like what happened. In terms of eukaryotic cells that can make their own food, the idea also goes into that afterwards. This cell engulfed another prokaryotic cell. Now, this prokaryotic cell have the ability to do photosynthesis. So the larger cell, again, rather than breaking the cell down for food, they allow it to live, and in exchange, another mutually beneficial arrangement has been made so that it provides food and shelter for the smaller guy, and in return, the smaller guy, now it's chloroplast, actually produce energy for the cell. Okay? So that's the basis of this endosymbiotic or endosymbiosis theory. You will learn more about this in grade 12 in more detail, especially when you're learning about cellular respiration and photosynthesis. But one of the many evidence in terms of like, hey, this theory sounds nice, but what are any evidence to back it up? One of the evidence to back it up would be um, the ribosomes. The ribosomes. So if you guys remember in eukaryotic cells, how do eukaryotic cells ribosomes different compared to prokaryotic cell ribosomes? Anyone happen to remember? Yes?
One's like a loop and the other's a helix.
Sorry, say again?
One is a loop and one's a helix.
That's the genetic material.
Oh.
Ribosomes. Anyone remember? Yes.
Eukaryotes have 3 subunits and prokaryotes have 2.
Not quite.
The size 170S, 180S.
Good. So the prokaryotic ribosomes are smaller, 70S. Eukaryotic ribosomes are bigger, 80S. And when you look at the mitochondria and the chloroplast, if you look inside, they have their own they have their own ribosomes, and the ribosomes found inside the chloroplast as well as the mitochondria are 70S. Okay, just like a prokaryotic ribosome. They also have their own genetic material. Okay, so in some ways they have their own like replication cycle that's independent of the eukaryotic cell itself. Okay, so that's a brief kind of talk in terms of the evidence of endosymbiosis or endosymbiotic theory. Okay, any questions? So conceptually, that's the heavier bit of today's lesson. Now, let's talk about the roles. Okay, these are the roles of protists in the environment. Okay, they do photosynthesis. Okay, they do photosynthesis. They're essentially the basis of most aquatic food pyramid. Okay, water pyramid. They are protists. Okay. Also, a lot of the pathogens, a lot of the human pathogens, are in the kingdom of protists. So, malaria, we're going to talk about it later on, is a parasite that is in mosquito as well as it will spend some time in mosquito and spend some time in humans. That is in the kingdom of protists. Beaver fever, that's a nickname for it. It's also another parasitic disease. Okay. So these are some common examples of pathogens, and these other ones are also made from protists. Okay, are we good? Now, because the kingdom is very, very diverse, and in some ways, if you look here, this diagram, there's many, many different groups. So we're going to be focusing on 3 major ones. Okay. We're going to focus on 3 major ones as an example to how diverse they are. So this one is paramecia or parameciums. Okay, they are single cell organisms. They're single cell organisms. They don't have a cell wall. They are mobile, and they are heterotrophic. Okay, so single cell, heterotrophic. And it's very mobile, don't have a cell wall. So here is a quick video of, you can see how these parameciums in action. So no, there's no sound to it, okay? But as you can see here, this paramecium can move around by the movement of the cilia around it. Okay, so it can move around through the medium. It is a live paramecium. Okay, And this thing here is later on it's going to start looking like beating like a heartbeat. You see there are live movements in here, and this vacuole will open, like get bigger and close, which is really, really cool. Not entirely sure what it is doing, but we in the past in this school, a lot of times we'll order in different protists that you can look at under the microscope. So hopefully we're going to try to do that after our bacteria lab first. Okay. Any questions? Okay. All right. The second one here, Eugenia. Okay. So this one here, it is another protist. Now this one, it has chloroplast inside. So in this case, it can do photosynthesis. It is autotrophic. Okay, it is autotrophic. In some ways, it is, if I remember correctly, it's like an algae. Okay, it is an algae. There are a lot of structures that are similar. Okay, you also have contractile vacuoles. You have nucleus and the nucleolus, but you also have a lot of different other parts. Okay. Now this here, now you can see here, I'm going to pause this here. You see these like the little green blobs inside here. A lot of those are the chloroplasts. Okay. Yes. Okay. And you see the flagellum that's beating and moving. That also helps it to move, but it doesn't appear to have a cell wall. Okay. It doesn't appear to have a cell wall. Yes.
Paramecia was. Paramecia was heterotrophic.
Yes.
Thank you.
Now, amoeba. Amoeba is another very classic protist. Now, this one here, it changes its shape a lot. A lot of its movement is done by changing of the shape. You have these thing called pseudopodium, podium. That's, I think pod is like related to foot. And so in this case, you basically have a lot. It will expand and kind of move around by changing it. It will also use this to engulf other cells. And in this case, in this video that you will see, you'll see an amoeba eating two parameciums.
So it moves by changing its shape?
So you see like it's extending a pseudopods around the two paramecium. These are the two paramecium right there. I'm going to turn down the volume. So it completely engulfed this now and put it into a vacuole. Now something is happening, and certainly the paramecium is not liking it. And movement seemed to have stopped. Okay. I remember, yes. Pardon?
Was that sped up?
I believe so, yes. Although I remember, is it last year? either Miss Lam or Miss Collin told me, like, when you're doing this lab, be careful. People don't mix up their pipettes, because apparently, like, one year someone mixed up the pipette. So they put either the amoeba into the wrong jar, which contained the other protists, and ends up that all the other protists got eaten. So do be careful, okay? When we're doing that, be careful. Any questions? Now, in terms of life cycle of protists, it is also very different and diverse. So the first two, asexual reproduction, this will be mostly for single cell or unicellular protists. Okay, so if it's unicellular protists, binary fission or conjugation, like a bacteria. If it is a multicellular protist, what they would actually do is they would actually mix. They would do sexual reproduction, but they would mix specialized cells called gametes. Okay, gametes are what we call specialized sex cells. So, like sperm and eggs in mammals. Okay, so gametes are sex cells, like for example, sperm and eggs in mammals. And we'll look at two different protists as an example that they would do. They would form gametes as part of their life cycles. Okay. How many of you guys are done here? If you're done, put up your hand.
Is that an M or an in?
I am.
Like, was that
in? I am.
Okay.
In. Okay. Now, you guys in grade 10, you guys learn mitosis, but you have not learned the process of meiosis, right? Yeah, you haven't learned this. You will learn this later on this year in grade 11. I'm just gonna write it here. So on this slide here, okay, it's the same different slide here. They said meiosis. Meiosis is the process of making gametes. Okay, it's a process of making gametes. It's very similar to mitosis. Essentially, it's almost like mitosis, but you do it twice. Okay. So it's a process of making gametes. Now, okay, can I move on? Or can I flip back? So let me flip back to here. Now, when you look here, you see like something about sperm and something about egg. And you see this N and 2N, okay? Any one of you guys remember what 2N might stands for? Diploid might stand for? No? Okay, so you guys, you learned it from, okay. Were you in this school last year? Yeah. Who was your science teacher last year in grade 10? Did you say good question?
Mr. Qureshi.
Oh, last year? He wasn't here last year. Last semester? Oh. Okay, so in this case, now let's talk about diploid. So in this case, mitosis, let's use human for example. In humans, how many chromosomes are there in a regular cell? Let's see anyone recall.
26 pairs.
Say again.
26 pairs.
Close.
Mhm.
23 pairs. Thank you. It's 23 pairs. The reason why we say 23 pairs rather than 46 chromosome because half of them are from mom, half of them are from dad. So that's why we usually like to say 23 pairs. Okay. So when we're talking about diploid cells will have all the chromosomes that it normally has. Different animals or different species have a different number. Okay. So not all species have 23 pairs. So diploid means diploid means it contains all the chromosomes from both mom and dad. Okay? From both mom and dad. So diploid means it contains all the chromosomes from both mom and dad. When you say haploid means it contains only half of the chromosomes. Usually you see that in an egg cell or a sperm cell. Okay. We'll go into more detail on this when we're in the genetics unit. We will learn about all two major phases of mitosis and actually 8 small stages, and you will learn how to draw them. Okay. What's the question?
It says egg from, isn't that the male
Female.
one? That's female?
Yeah, this is female. This is male. If I remember correctly, this was supposed to be like represent the shield and the spear of the Roman god Mars, and this is supposed to be like the mirror of Aphrodite. If I remember my Roman mythology well enough. Okay. Yes.
What's an example of something only having half of the chromosomes?
Like an egg cell or a sperm cell. So if you think about it, an egg cell and the sperm cells come together. You have a fertilized embryo that becomes a baby. Okay, so the fertilized egg or embryo, the baby have 46 chromosomes, 23 pairs. So half of that, 23, were from the sperm cell from dad. Half, 23 of those would be from the egg cell from mom. Okay, so haploid. Yes.
So wouldn't that like feature as like diploid or whatever? Wouldn't it just get more and more and more chromosomes?
No, and when you learn the process of meiosis, you'll realize why. Yeah. Are we good? As I said, it's like mitosis, but twice. Okay, so in this case here, you can see that. Let's start with the fertilization of the sperm and egg for this life cycle. This is a brown underwater algae. Okay, agar. So in this case, start with the fertilized egg, which is called a zygote. It will go through a different phase. It will start developing sporophyll, what you call sporophyll. So at this point, which is like what you see, it reminds me of kelp. I'm not sure if it is kelp. It will start producing the structure called sporangia. And in sporangia, inside, you will start the process of making sperm and egg. Kind of like, if you want to think about it, yeah, it's very similar to making the gametes in flowers. Okay, you will see Angia. It's also angiosperm. Is actually means flowering plants. Okay, so flowers too have sperm and eggs, pollen, and whatever that term is inside. So you start making the two smart, the male gametes and the female gametes, and then later on they would come together called do fertilization. Okay? Having a specialized cells to do sexual reproduction would make sure the genetic diversity being bigger, being better. Okay. Any questions? Yes.
How does it ensure that the genetic diversity is bigger?
Because in a similar way, in a similar manner, it's almost like shoveling a deck of cards. It just shuffled the decks better. In some ways, in a very simplistic answer, it's like you shovel the deck much better.
So, like those cells just shuffle the deck better than
If you're using sex cells, the process of meiosis, yes, than just conjugation. We good? Now this one, malaria. Plasmodium. Plasmodium is a genus of three different protists parasite that causes malaria. There's actually three different species. Okay. Now, what we want to show you here, not only because malaria is a big health hazard, it's still ongoing, definitely, and it is a disease that have a really wide scope around the world. But at the same time, it's also interesting because you see that in its life cycle, you see it in two different species. Half of its life cycle is in a mosquito, and then in the other half of the life cycle, it's inside human liver and red blood cell. Okay, so in some ways, it's an interesting one to look at and to study, of course. So let's go with this part here. Okay, a mosquito bit someone who has malaria. When a mosquito sucked the blood of the person who had malaria, they would ingest the red blood cell. Now, inside the red blood cell, that's where the parasite lives. That's where the plasmodium lives. And in this stage here, you have the gametes. Okay, you have the gametes of the plasmodium, and inside the mosquito, that's where the gametes start creating zygote, like a fertilized egg. So the earlier stage of the plasmodium, it is in the mosquito. It is inside the mosquito. Now this mosquito that is infected with plasmodium bit someone else. Now, when the mosquito bites someone, they do spit out some saliva in there, and that saliva contains a certain enzyme to help to help blood not clotting. Okay, because normally if we have a cut, our blood will clot to limit the blood loss. Mosquito saliva has some sort of enzyme to stop that or slow that, so that they can have a blood meal. So when that situation happens, the plasmodium will go into the blood. Now, all the human blood will go through the liver. The liver is a place where the blood gets detoxed. So inside the human liver, there will be some of the stages where the plasmodium will be living, and then it will go into the human red blood cells. Okay, again, Plasmodium will live in the human liver as well as inside the red blood cell. Okay? And in this form around here, this is also the form that they will start producing their gametes. We will look at this part here in terms of the red blood cell and plasmodium or malaria. You'll see this again later on in the units of genetics because it has some relationship to another genetic disease or condition called sickle cell anemia. Any questions? But we're not going to talk about sickle cell anemia right now. This is just to show you how an example of a life cycle of a protist that exists in 2 different species and 3 different types of tissues. Yes?
What's the incubation period for malaria?
I have to look that up. I don't remember. Yes? Yes. Any questions? Yes.
What was Sherrod's question? I didn't hear.
Oh, what's the incubation period for malaria? So I said I have to look it up. Don't quite remember. We good? All right. What time does class end? 40. Okay, so we have a little bit of time. Now, what I want you guys to do, come up here and pick up a handout. These 2 tile are identical. I just want to limit crowds. Okay.
Where's mine?
Oh, I got one.
Yay. Culturing bacteria. This reminds me of,
Why physics?
because we get like really big packages with like a bunch of work.
A bunch of labs?
No, not lab. It's like practice.
Oh.
But they're usually this big.
Oh, that's different.
Okay, yeah.
Yeah. Physics, chemistry, engineering students love. Oh, you found. Okay, thank you. Yeah, physics, engineering students love practice sets. Practice sets. So, we're doing a bacteria lab. This lab will do it tomorrow. Okay. Now, this lab is a multiple day lab. I will be assigning the groups. I do not remember. I will have to double check with Miss Lamb because I want to know how much material we have, the material constraint. But I will be assigning your groups. Now, this lab is a multiple day lab. So don't put things away yet. Please look up here. You might want to jot down some stuff. So this is a multiple day lab. As I said one more time, tomorrow we're only doing part 1. Tomorrow we're only preparing the nutrient agar. So when you are growing bacteria, a lot of times you see people have a petri dish and have this layer of like orangey thing that they swipe and grow the bacteria on. That's nutrient agar. And tomorrow, you guys are going to prepare your nutrient agar dishes. Okay. So as you can see around the room, Miss Lamb's class, they have their stuff there, and they will be cooking later on today in their class. Tomorrow, we will be also doing the cooking too. It's very, very important that you are here tomorrow, simply because tomorrow we're preparing the nutrient agar. And sometime next week, we'll be doing the other 2 parts of the lab. So if you're not here tomorrow, then it will be tricky for you to kind of like, hey, how are you gonna have plates for the future steps? Okay, so make sure you're here. Also, even though we are not using a live flame, but we're heating stuff, we're boiling stuff. If you have long hair, please bring your own hair tie. I mean, if you don't bring yours, the only thing I can provide would be like elastic bands. Okay? And last I remember, it's not that comfortable. Okay? Yes?
I don't have to.
No. Unless you like you're not like have a danger of like dipping over and like have hair getting into whatever it is. Okay? Also, loose clothings would you should try to avoid that. So wear layers. Wear layers. Okay. So you don't have like baggy sleeves that can knock things off. Also, another thing, because the steps of this cook is going to be a little tricky, I will ask you to read the procedure for part 1. Read the procedure of part 1. You do not need to do the questions, okay? You do not need to do the questions tonight. I want you to read the steps and procedure for part 1. I will give a pre-lab quiz tomorrow to just check that you have read it, okay? Last year, a number of groups, when they're doing their part 1, because one thing they didn't do quite correctly, a number of groups have to restart the whole thing, and the whole place smells not so nice. Okay, so try to read this. Okay, so your primary homework tonight would be to read the procedure. Okay, and prepare for the quiz. And the secondary homework will be doing the Protist textbook questions. All right. Yes. The homework, yeah. Okay. Any questions? Okay, yes.
What is a J cloth?
J cloth is a type of cloth. It's inside the box that you guys will use tomorrow. Okay. Any questions? Okay, so where is my